Students, let us continue the lesson, the principle of inheritance. The principle of inheritance, on which we have to talk about the genetical basis of ABO blood system. We have seen already the biochemical analysis. Today we have to, that is about genetical basis. In 1925, a person by name Bernstein observed three alleles. That is, in three different rows of chromosome number 9. They are responsible for, that is a blood group system. The alleles are named as LA or LB or LO. Now the L refers to Carl Landsteiner, honoring Carl Landsteiner. We are using the letter L. So this is allele A, allele B, allele O. It may also be represented as IA allele, IB allele, IO allele. So we have two forms of representation, either in the form of L, A, B, O, or in the form of IA allele, IB allele, IO allele. So here I mentioned L stands for Landsteiner. There I stands for the gene isoheme-agglutinogen. Isoheme-agglutinogen gene. Isoheme-agglutinogen gene. So in the book it is given isoheme agglutination. It's not so. Isoheme-agglutinogen gene. So I stands for isoheme-agglutinogen gene. So these are the three alleles located in chromosome number 9 that was proposed by, that is Bernstein in 1925. Now let's see, what is the role of these alleles? IA and IB are coding for enzyme. One enzyme, IA, IB, these are coding for enzymes. What is called transferase. This is the enzyme encoded by these two alleles, A and B. Now IA normally just this gene encodes or produces the enzyme N-acetyl galactose transferase. This is the enzyme produced by allele A, N-acetyl galactose transferase enzyme. This enzyme is responsible for the union of a substance, namely N-acetyl galactosamine, to a precursor, what is called H substance. This is a precursor, the basic substance. So the allele produces an enzyme, N-acetyl galactose transferase. That enzyme is responsible for the union of this N-acetyl galactosamine to a substance, what is called H substance. Normally this is called actually Bombay antigen, so as to form what is called antigen A. So this enzyme catalyzes the formation of antigen E from N-acetyl galactosamine, that is a substance, that is H substance, H antigen, to form antigen E. So there is a role of that enzyme. That enzyme is normally coded by what is called A allele. Similarly, this B allele, it normally responsible for transferring what is called galactose. That is to H substance to form antigen B. So this is the reaction catalyzed by an enzyme what is called galactose transferase. This is what is called actually transferase. Generally, the enzyme is called transferase. The allyl allele produces N-acetyl galactose transferase. Now, the B allele, it produces what is called galactose transferase. It produces what is called galactose transferase. So, Ia allele is responsible for the production of N-acetyl galactose transferase. That enzyme normally catalyzes the reaction, namely that's N-acetyl galactose amide plus H substance to form antigen A. And now this B allele produces only that is galactose transferase. That galactose transferase normally that is actually transfer the galactose to H substance to form antigen B. So anyway, commonly the enzymes are called transferases, but we have two different what we have enzymes. N-acetyl galactose transferase for catalyzing the formation of antigen A and galactose transferase for catalyzing the formation of that is antigen B. So this is what we have, the formation of what is called antigen A, antigen B. Whereas IO, this is the allele. This allele is called as a null allele. This is called as a null allele. This is called as a null allele because it doesn't have any code either for NAG, N-acetyl galactose transferase, or for galactose. Doesn't have any code for the production of enzymes for transferring NAG, N-acetyl galactosamine, or galactose to the corresponding what is called H substance. That's why it's called as a null allele. That means it doesn't code any enzyme, and so that it is called as a null allele. And normally, I mentioned already, it doesn't have the ability of just transferring NAG or galactose either to H substance to form antigen A, either to what we have or to H substance to form antigen B. So this is what we call the genetical basis. So the main idea here, the alleles are responsible for the production of a particular enzyme, and those enzymes are responsible for transferring a particular substance to a H substance to form antigen A or antigen B. And O allele doesn't have any, just actually ability to produce a transferase enzyme. So there is no transfer of such NAG or galactose either to A, either to H to H substance. So there is no formation of any of the antigens. So this is the genetical basis for what we have actually the ADO blood system. Now we have actually, in addition to human beings, in some animals also we have the blood system. For example, in the case of there is what is called greater apes like orangutan. This is what is called a greater ape. Or we have chimpanzee. Or we have, for example, just we have gibbon. These are all greater apes. In the case of chimpanzee and then what we have chimpanzees, we have a blood system having A antigen. In the case of orangutan, this is another type of greater ape where we have antigens A or B or AB are present. So these are examples other than human beings, animals having the blood group system. In the case of, for example, Platidaria, there is New World monkey. This is called, say, an example of Platyrrhina. The meaning for Platyrrhina, they have flattened nose, rhinology, the study of nose. Platyrrhina, flat nose. And this New World monkey is having a substance similar to B antigen but not identical to B antigen of human beings. So this New World monkey, Platyrrhina, has actually an antigen similar but not identical to what's called humans, just like B antigen. In the case of cat also, we have three blood group systems. As in the case of human beings, three blood group systems. Same in the case of cat too. Now, Under this category, we have now one more question. What are secretes? What are secretes? What are secretes? Now, normally, we are talking about the blood group antigen. That means the antigens found in the blood groups. In some individuals, the antigens A or B are present in the saliva, in the tears, even in the semen, even in the milk of mammals, or in the case of gastric juice, or in the case of urine. So, the antigens, blood group antigens also present in saliva secretions, tears, semen, milk, gastric juice, urine, etc. Such individuals having the antigens A or B in these materials or in these secretions are called as secretors. Individuals with antigens in saliva, tears, semen, milk, gastric juice and urine, these people are called secretors. So that's about what we have actually AB blood system. Now we have to go for the next one, what is called RH factor. What is RH factor? I explained this one already in 11th standard. Anyway, little bit I want to go further. What is RH factor? What is RH factor? Now in 1940, two people, Carl Landsteiner and Alexander Wiener observed a special type of antigen formed on the surface of or basis of a monkey, what is called Asian monkey, rhesus monkey. The name of the monkey is Macaca rhesus. The species name rhesus. So, the antigen that was reported from the monkey was given the name that is also called RH factor by taking the first two letters of the monkey. This RH factor is also called as D antigen. This is also called as D antigen. So what is RH factor here? The D antigen, that is in the immunogenic D antigen. Immunogenic D antigen means the one which provokes immune response. So RH factor refers to the immunogenic D antigen of the RH blood system. Immunogenic D antigen. I am repeating the word immunogen. The word another name given for antigen. We give different synonyms, but I don't want to take that one. So anyway, the D antigen is also called the immunogen. So the D antigen, so the RH factor is immunogenic D antigen of the RH blood system, reported in rhesus monkey. The same antigen is also present in most of the human beings on the surface of or basis, in addition to the normal antigens A and B. For example, nearly 85% of the human population is having this antigen, what is called the D antigen. So, based on this one, we have two groups of people. People with D antigen. So, why we are using the letter D antigen? Because the RH factor is inherited as a dominant factor. The RH factor is inherited as a dominant factor. So the gene D, a dominant gene, is responsible for the production of the RH antigen, hence called as actually D antigen. 85% of the human population has this D antigen on the surface of our basis. The 15%, the remaining 15%, they do not have this D antigen on the surface of our basis in addition to normal antigens A and B. So the presence of A or B or absence of both, that is different. So in addition to those antigens A or B or both, we have this antigen present on the surface of all species of human beings. So 85% of the population having this big antigen on the surface of all species in addition to A or B antigens or both antigens. Now the remaining 15% of the will have no origin antigen or D antigen in addition to what we have A or B or both. Now let's go forward. Now we have this origin antigen has a specific what is called a genetical basis. I mentioned already this is nothing but what's called immunogenic D antigen of the RH blood system. The name is given because it is inherited as a dominant factor, and also the name is given RH because it is first known from the rhesus monkey, Macaca rhesus, an Asian monkey. Now, what is the genetical basis of this RH factor? We already discussed this one in 11th standard, that's why I am going a little bit faster, but you can understand what I am saying. What is the genetical basis of RH factor? Genetical basis. We are not studied in 11th standard. We studied only what is RH factor, what is impact on human beings. Here we have to go for genetic analysis. Then what is the impact on human population? How can we prevent it? These are all the things. So genetical basis. So there are two opinions. Two groups of people have two different types of opinions. One is what's called Aurora race and Olive Fisher hypothesis. This is one hypothesis. Aurora race, Olive Fisher, Aurora race and Olive Fisher hypothesis. So according to these two people, they have used what is called CDE nomenclature. Their nomenclature is called CDE nomenclature. This is also CDE nomenclature. CDE nomenclature. The most hypothesis common in use today is nothing but CDE nomenclature. Actually, what it means CDE nomenclature. According to these two people, there are three different pairs of alleles present in three different loci of chromosome number 1. So I am taking for example, this is the homologous pair of chromosomes. That is what we have first pair. This is the first pair of chromosomes. So in the first pair of chromosomes, I am drawing two chromosomes which are homologous in nature, having three different loci. Now, three different loci have three different pairs of alleles. Now, here we have capital C or small c. Here we have capital D or small d. Here we have capital E or small e. So, this is the locus, the point, where we have the particular gene is located. Either we have C or small c, the recessive gene, or a dominant gene capital D, or recessive gene small d, or just actually the dominant gene capital E or small e. We don't have both at the same time. Either we have capital C or small c, not both. Likewise, either we have capital D or small d at that locus. Similar is the case here also. We have the same chromosome. We have either small c, small d, that is what we have small e, but not both. So this is what we got homologous pair of chromosomes. Suppose for example, I mentioned CDE, actually nomenclature. So these two are the homologous member, the first pair, we have three loci, where you have three different pairs of alleles. An individual may have actually three genes. For example, if you are taking in one chromosome an individual having all the three genes dominant. If you are taking the heterozygous condition, we have in another homologous member we have what is called the recessive genes. So this is one type of what is called actually homologous pair. I can represent this one C, D, E and C, D, E. This is one nomenclature. Sometimes we have in both the chromosomes, actually we have that is both recessive genes in both the chromosomes, like this. But in some cases we have a chromosome is having that is like this, or we may have, for example, C capital C, that is small e, then small c capital D, small e, like that. So this is a combination of homologous chromosomes. In some cases we have an individual in both the chromosomes having dominant gene. So this is the analysis. This is one homologous chromosome, another homologous chromosomes. Now this is called CDE nomenclature. So the OH family is controlled by 3 different alleles, not a single allele, present in 3 different rows. Now, the D allele is the dominant allele when compared to, that is capital C, capital D, or small c, small d, small d. So D is the dominant allele. If you have actually an individual having dominant gene D, he is having what is called antigen OH antigen. And suppose here in both the chromosomes we have no dominant gene. The person doesn't have antigen OH. Now in the third case we see in one of the homologous member we have dominant gene D. So this person has the ability of producing OH antigen or D antigen. So he is actually having OH antigen. This next case also we have DG, the dominant gene. So in these combinations, at least one dominant gene D is present that resulted in the production of what is called actually that is D antigen. Such people with D antigen are said to be RH positive. People with RH antigen are said to be RH positive. And people without this antigen are said to be RH negative. RH positive with antigen RH, RH negative without RH antigen. For example, here I am taking this one. So these people having the genes all recessive are considered as actually recessive individuals and they do not have the ability of producing. In all of the cases, you can see that at least one dominant gene, either in one chromosome or both the chromosomes. Here both the chromosomes. Here one chromosome. Here one chromosome. Here one chromosome. They have the ability of producing antigen. So, this is what is called CDE nomenclature proposed by R.R. Rees and R.A. Fisher, and calling them 3 different alleles which are present 3 different loci linked together in chromosome number 1 responsible for the production of RH antigen. This is what is called R.R. Rees and R.A. Fisher hypothesis. Common use today is this type following we are, and CDE nomenclature, that is called CDE nomenclature. Now another genetical basis proposed by A is Wiener, Alexander Wiener. So that is called Wiener hypothesis. Wiener hypothesis. According to Wiener hypothesis, he proposed there are 8 alleles. 8 alleles. There we have 6 alleles, but we have what is called 3 pairs of alleles. Here 8 alleles. These 8 alleles are located in one locus, same locus, not different locus. There we have the 3 pairs of alleles are located in 3 different loci closely linked in chromosome pair 1. But Wiener hypothesis, he proposed there are 8 alleles. All the alleles are located in one locus. Now the alleles are R1, R2, just actually R0, or Z. These are all actually the 4 dominant alleles. What are the recessive alleles? R, R1, R2, RY. These are the recessive alleles. So R1, R2, R0, or Z. This is Z. The recessive alleles are present in the same locus. So individuals with any one of the dominants, for example R1, that is R, or R2 with R2, R0 with what is called RY, any one we can have. So or suppose an individual is having the genotype of capital or small r, then he can have the ability of producing the antigen. So this is heterozygous condition. Or if you have, for example, homozygous individuals. So this is also homozygous condition or 0. Now this is homozygous condition. All these individuals produce what is called the antigen, D antigen or RH factor. But these individuals, when you have the recessive individuals in homozygous condition, either like this or so these are the homozygous recessive individuals. They cannot have the ability of producing the antigen, D antigen or RH factor, so they are said to be RH negative. So according to what is called just RR race and RR fission, 3 pairs of alleles, 3 different laws, okay, and the combinations I mentioned. The D gene present, we have the production of RH factor. Here, according to Wheeler, there are 8 alleles. All the 8 alleles are found in one locus of different individuals. Suppose, for example, I am taking one individual. Here, locus 1 locus. Here, R. Or in the case of heterozygous condition, R. So here we have actually same locus all the alleles. Suppose you are considering, for example, the 6th locus. Either capital R1, R2, R0, or Z, or R1, R2, Y, all present in the same locus. So this is homozygous condition, this is heterozygous condition. In all cases, when you have one capital R is present, then it is being expressed. So that we are getting what is called the antigen. If it is both, for example, this is a homologous pair, we have R. This is a homozygous recessive and negative producer. So all present in the same locus only. This is the meaning for that. That's why it's an example for multiple allelism. So not only in vivo blood group but also the RH factor is coming under the multiple allelism. The reason for that one, they have more than 2 alleles affecting the same character. What are called the RH factor. So this is the genetical basis. If you are unable to follow this one, let us have a discussion of the story of this. So with this I conclude. I pass on to the next part of this RH factor. What is the impact of this RH factor? What is called incompatibility. Suppose, for example, so we have RH positive and negative. So for example, A positive persons have A antigen and RH factor. B positive persons have actually B antigen, then what we have RH antigen. Likewise AB positive and O positive. And if you are taking negative, some people have A antigen but no RH factor. Some people have B antigen but no RH factor. Some people have both AB antigens, no actually RH factor. Likewise O. So such people are said to be A negative. They have A antigen but not RH factor. B negative, they have B antigen but no what is called actually RH factor. Accordingly, we have AB negative, O negative. Now the most, I mentioned already 85% of the people have RH positive. The most common type is O positive. The most common type is O positive. Next comes A positive. Next, so the most common type of blood group is O positive. Then next comes A positive, then B positive, and finally AB positive. So I mentioned already A positive, A antigen with RH antigen. Now let's go further. In blood transfusion, what will happen if RH positive blood, so I am taking what is called RH positive blood. It may be either A or B or O or A. When it is transfused, so this is person A just it is being transfused to person B. So he is having RH negative blood, irrespective of what is called A or B, AB or O blood groups. The blood is transfused from RH positive to RH negative. What would happen? Is there any effect? Now, one of the actually principle used in blood transfusion, the donor should not contain specific antigen against specific antibodies produced. Specific antibodies produced. Now, actually, unlike A, B, AB or O blood groups, they have antibodies, either A or B or no antibodies. They are called natural antibodies. They are called natural antibodies. The antibody A, anti-A, anti-B antibodies are natural antibodies present in ABO blood system. They are called natural antibodies. They are of what is called IgA type. So I repeat once again. This anti-A, anti-B antibodies are IgA type. Chemically, the antibodies are immunoglobulin. We have 5 different types of antibodies: IgA, D, E, M, and G. We'll discuss later under immunology. Now, this one cannot pass across placenta. But for RH factor, there are no natural antibodies. No natural antibodies. No natural antibodies for RH antigen or against RH antigen. But we can stimulate the formation of RH antibodies, or the anti-D antibodies. So, the RH antibodies, otherwise called anti-D antibodies, because working against what is called antigen D, just like A, anti-A, anti-B, like that. Now, when RH positive blood is transfused with RH negative blood, here there are no natural antibodies. But one of the principles used in blood transfusion, the donor should not contain specific antigen against what is called antibody present in the recipient. The donor should not contain specific antigen. But here you see that this blood contains RH antigen. So when RH antigen enters here, it stimulates the production of what is called RH antibodies. So that what is happening, reaction occurs between anti-D antibodies or RH antibodies and RH antigen, leading to what is called the clumping of blood cells. So we cannot donate RH positive blood to RH negative. This is because the antigen enters into the recipient's body, person B, and stimulate the production of antibodies, not instantly, but slowly. And these antibodies are working against RH antigen, so that what will happen, we have the clumping of blood cells. Suppose a person B is donating blood to A. I mentioned already one of. So when person B who is having RH negative blood, when this blood is transfused with a person with RH positive, nothing would happen. Because I mentioned the principle, the donor should not contain any antigen. So they can give a blood. So here RH positive person cannot donate blood to RH, can donate blood to RH, RH positive person cannot donate blood to RH negative blood because of the development of antibodies and clumping of blood cells. Whereas RH negative person can donate blood to RH positive person because no antigen is present, as it follows the principle of blood transfusion. The principle used, I mentioned already in blood transfusion, the donor should not contain antigen. As this RH negative person, the positive blood is donated to RH negative. Now the anti-D antibodies are developed by the B lymphocytes after some time. So reaction occurs between antigen and antibodies and clumping of blood cells occur. So we cannot donate RH positive to RH negative. But RH negative person that donates blood to RH positive, no reaction occurs in the RH positive person because this negative person has no antigen to stimulate the formation of antibodies. So no reaction,